Welcome to an overview of the integumentary system. So let's get started by orienting ourselves with the anatomy of the skin. So our skin is actually our largest organ in the body. Accounts for about 10 to 15% of the total body weight. And it is a complex organ with many functions important for homeostasis. There are two layers to our skin, the epidermis and the dermis, but we have a third layer that is frequently talked about, the hypodermis. The epidermis is just this upper area of the skin with the dermis composing most of the skin. All three together are a cutaneous membrane. Embedded in the skin, we have hair, glands, we have blood vessels that are running through. This is the erector pili muscle that makes your hair stand on end or gives you goosebumps. We also have lots of sensory receptors within the skin. The skin has multiple functions. Protection, it protects us from mechanical trauma, pathogens, and the environment is most obvious. So this protection is its most obvious function. We have sensation, so perceiving changes to the external environment, whether it be touch, temperature, sensing, all of that is done via the skin. We have thermoregulation. This is mainly done through our sweat glands. Excretion, so with that sweat we actually excrete waste products, much like what happens with our kidneys. Skin plays a critical role in vitamin D synthesis. This is done in cells found deep in the epidermis that convert vitamin D from its inactive form to its active form. It's also a major blood reservoir. That's the dermis's job. As you'll learn shortly, the epidermis is actually an avascular tissue, meaning A means no vascularization in our epidermis. And so here is a feedback loop. Remember, feedback loop is one of our main core concepts. And so when our body temperature gets too high, thermoreceptors sense that the temperature is too high, communicates with the brain to set off a series of events to cool us down. Causes vasodilation of our blood vessels, which rushes blood. That's why we get flushed when we get hot, because blood rushes to the surface. That way it can release the heat. And also we secrete sweat onto the surface of the skin. And if you recall back to the properties of water, that high heat of vaporization allows the water to carry away a lot of this heat that is being released via vasodilation. This brings our body temperature back to normal temperature. Same thing when we get cold. If we get too cold, thermoreceptors sense it, goes back to the central nervous system, it detects it. We had vasoconstriction, so conserving the heat, so the blood is not towards the surface, keep all the blood in the core. And that's why when you get too cold, like with frostbite, your extremities start to actually die because they lack blood flow. And so this brings our body temperature back up to normal. So let's look at the epidermis in more depth. The epidermis itself is made up of 4 layers, no matter where you look at it. But we have this other layer, the stratum lucidium, that we only find in thick skin. And thick skin is predominantly found on the palms of your hands and the soles of your feet. So this upper layer is our thickest skin, and this is the part that sloughs off, makes up a lot of the dust that you see around your house. So the stratum corneum is our most exterior of our skin layers. Then we come down in that next layer, the stratum lucidium, and again this is only seen in thick skin. And so this is the photomicrograph where you can see it. Here's our dead and dying, or they're dead cells. The next layer is the stratum granulosum, And here you can see that the cells actually still have a little bit of a nucleus and they're kind of spotty. then the stratum spinosum, and the stratum basialis. And the stratum basialis is just this one layer down here. It's usually a single layer, maybe two layers. And here you can see a dividing keratinocyte. So the stratum basialis is where we have all of our cell division. This is where the skin is constantly renewed and replenished. And so the stratum basialis undergoes active mitosis, and it helps to replenish the cells that have died and sloughed off out here in the stratum corneum. You can see that as the cells move away from the stratum basialis, they get, as I pointed out earlier, kind of fragmented. The nucleuses start to degrade. You can look over here on the photomicrograph. Here are these dark blue spots. You can see how healthy these cells are with a strong staining nucleus. But the further you move away, they start to die. And if you recall, I pointed out earlier that the epidermis is avascular, so there is no vascularization in the epidermis. And so for the epidermis to get its nutrients and its oxygen, everything that the cells need to undergo cellular processes, they have to get it via diffusion from the dermis. So all the vascularization of our skin is in the dermis. And so they have to get it for diffusion. And so the farther they get away from the dermis, they start losing the ability to get nutrients and oxygen and all the things they need to carry out their metabolic processes. And so they die. So the further away you get from the dermis is why the cells die, and that's why the outer layers of your skin are actually dead. Other cells that you'll find in here, you have the dendritic cells. These are also known as our Kupffer cells. You have Merkel cells. So Merkel cells are a sensory receptor cell that you find in the epidermis. And you have a melanocyte. Melanocyte is what gives our skin its pigmentation. And so when you are exposed to the sun and your skin gets darker, it's because the melanocytes are producing melanin to deposit into the skin. It also gives us all of our various tones of skin. So melanocytes make a pigment that we'll talk about a little more, melanin, that gives skin its color. And here is a simple trick to remember them. Brilliant studying gives loads of confidence. So that's going from the most deep area to the most superficial. So your basal, your spinosum, your granulosum, your lucidium, and your corneum. Make sure you always put stratum in front of that. Stratum corneum, stratum basiala. Make sure you put the stratum in front of that. And also remember, only our thick skin has the stratum lucidium. So if you get confused with which is the most deep versus the most superficial, think B as in basal as standing for the bottom. That is our bottom layer. And so how do our keratinocytes move to the more superficial area of our skin? And so watch this cell A. So cell A right here, actively dividing, and as it actively divides by mitosis, it pushes the cells up, and then so here A, and then it divides. B is pushed up, A stays to undergo mitosis again, undergoes mitosis again, pushes B even further up, undergoes mitosis again, pushes B even further up, undergoes mitosis again, pushes B even further up. So as the basal layer continues to undergo mitotic divisions, it pushes the previously divided cell towards the superficial layer of our epidermis, and as it moves further and further away, it starts to die. And so this takes a cell between 40 to 50 days to complete. So migration from the deepest strata to the stratum corneum takes about 40 to 50 days to complete. So your skin is basically replaced about every 40 to 50 days. And those outer layers are what slough off. As I mentioned before, our thick skin has that stratum lucidium. It is about as thick as a paper towel. So it has all 5 layers. Thick skin also does not have hair follicles. but it contains many sweat glands, and that's why you get sweaty palms, but you'll never have hairy palms, no matter what your mom tells you. Areas of the body that are not subject to such mechanical stress have thin skin, so that's the majority of the rest of the body. And so it only has the 4 layers, lacks the stratum lucidum. Each of those 4 layers is thinner than those found in thick skin, but it has numerous hairs, sweat glands, sebaceous glands. And this is where you'll get, and that's why you'll leave fingerprints with the sweat glands. When you get a callus, remember calluses tend to be in thick skin. And so the layers of the stratum corneum form either a thick or thin in response to repetitive pressures, but you tend to get those more in your thick skin, but you do because the back of your heels will get blisters too, or calluses in the thin skin. And so here is a comparison of our thick skin and our thin skin. So again, stratum corneum is much thicker in our thick skin than in the thin skin. Have the extra stratum lucidium in our thick skin versus the thin skin. So here is a comparison between thick and thin and how they vary. So I mentioned receptors, that we have receptors in our skin, and these are our majors. I pointed out the Merkel cell in the diagram before, and that's in our epithelial. It's associated with small neurons in the dermis. So the sensory receptor is actually in the stratum basialis and then is connected to a neuron in the dermis, detects light touch, discriminate shapes and textures, found in large numbers, especially in our fingertips, because that's where we're going to do textures and shapes and all those things. 2 other receptors that we'll find in the dermis are tactile or Mesner's, and these are going to be up towards the intersection of our epidermis and dermis, because again, they're going to do light touch. And so they're going to also be more numerous where we need sensation. But we have other called lamellated or Pacinian corpuscles. These are deeper into the dermis, down in that reticular layer of the dermis. And so they are going to be embedded, and they're going to feel pressure and vibration, so things that are stronger sensations. Here is the skin model that we actually have in lab, and so you can see all these structures. Here's thick skin on the skin model. See, the corneum's much thicker, and the little white layers only here, that's the lucidium. And then you have the other layers, the granulosum, spinosum, and basialis. Here's the key for our skin model. Here are the receptors that I had just pointed out in our previous. And so here, Mesner's and Pacini, you can see both here, Mesner's and Pacini. We can see our different glands, and you'll learn shortly that there's 2 types of glands. If they empty right onto the skin surface, they'll be esserin. If they empty into a hair follicle, they're going to be apocrine. And here's a sebaceous gland. So the sebaceous is more of a lobule gland. Here's our hair follicles embedded and the different layers of the hair follicle. Here's the adipose, the dermis, and the epidermis. So here's the key to the skin model, and we can find all those different structures that we've mentioned, the erector pili here, all in our skin model in class. Here is an older skin model. We can see a lot of the exact same structures. Again, the Pacini, the Mesner's, the esserin. I don't think that we can see the apocrine on this particular shot. Sweat gland, these are the sebaceous, the hair follicle, and thick skin versus thin skin over here. Stratum lucidium, this layer right here. So now let's talk about the dermis in more detail. So the dermis has more functions. It's our deep layer to the epidermis, provides a blood supply to the epidermis. As I mentioned earlier, the epidermis gets all of its nutrients and oxygen from the blood through diffusion from the blood that's in the dermis. You have the sensory receptors that we've looked at, and it also anchors the epidermis in place. Our dermis has 2 distinct layers. Has the papillary layer, which is this upper layer where you have these dermal papillae. And the dermal papillae are coupled with our epidermal ridges. And so this is what gives us our fingerprints, these epidermal ridges that are helped to be made by our dermal papillae. You have tufts of capillaries up in each of these dermal papillae. And then the majority of our dermis is the reticular layer. So this is this larger section of the dermis. Here's a photomicrograph of our dermis over here. And as I just mentioned, those epidermal ridges that are formed by the dermal papillae are what give us our fingerprints. And you have sweat pores. So that would be our Esserin sweat glands, because they empty directly onto the skin surface in all of these epidermal ridges. And that's why we leave behind fingerprints, because we leave behind a layer of sweat on anything that we touch. Our skin also has these tension lines. And so here are the tension lines across your body. Any good plastic surgeon especially is going to know the tension lines, because if you cut with a tension line, you're going to have a lot less scarring than if you cut against your tension lines. Because if you cut with the tension lines, there's no pulling and stress on it. But if you cut against the tension lines, and those tension lines are constantly pulling that incision back open. And so that would give us more scarring. And so in areas such as joints, reticular layer, deeper creates deep creases called flexure lines also. And so again, these are all of our tension lines across the body with the skin. All right, so let's stop this segment of our integumentary lecture, and then we'll pick back up with skin pigmentation. Let me know if you have any questions, and have a wonderful day.